The 11th chapter in the first PUC is transport in plants. Transport of minerals, water and all those things takes place in the plants by different processes like the diffusion, osmosis, all those things. And coming to the diffusion is a process in which molecules move from the region of higher concentration to lower concentration. It does not involve any semi-permeable membrane. However, diffusion is a slow process. It is very common in case of the gases and the liquids. The diffusion is very common, and it takes place in the different aspects, that is the gases, the liquids, in case of the plants. There is one more diffusion called facilitated diffusion. So one question so far not asked: the lipid soluble substances move faster across the cell membrane than the water soluble, which is very, very important. As I told you earlier, the lipid layers, the polar molecules like water doesn't cross through the non-polar lipids. Hence, the lipid move faster than the lipid soluble substances, than the water soluble substances. And for this, water has to transfer along with the salts. It requires some extra proteins. So the transport across the membrane, in which the proteins facilitate the transport by diffusion, which is a passive process. It doesn't involve the expenditure of energy. That is called facilitated diffusion. So these proteins, especially the proteins, not the membrane proteins, the other proteins which are present in the plant body, they act as the carriers, and these proteins are called porins, and there are about 8 aquaporins in the plant body. And facilitated diffusion is of 3 types. Here, these proteins allows diffusion of the two molecules, that is symport, and sometimes these proteins facilitated during facilitated diffusion allows the two molecules, but in opposite directions, called antiport, and single molecules in one direction called uniport. So code word for this, USA, uniport, symport and antiport. That is the facilitated diffusion, which doesn't require any energy at all. And coming to the active transport, takes place at the expense of energy. That is also facilitated active transport, in which membrane proteins, they carry the molecules across the membrane at the expenditure of energy. So this kind of facilitated active transport, that which is facilitated transport at the expense of energy, can be seen in 3 examples I will give. So the transport saturates, and then highly selective and require membrane proteins are the 3 proteins, that is facilitated transport, and requires ATP and uphill transport are the 2 active transports, but not facilitated transports. Thus, a column is given in the textbook about diffusion. No energy involved, and facilitated transport to 3, and then the all are, that is the transport saturates, highly selective, require membrane proteins, require ATP, uphill transport. All these are the active transport which takes place at the expenditure of energy. The water molecules, the water content in the watermelon is about 92 is only water. The corn absorbs water, about 3 liters of water per day, whereas the mustard absorbs water equal to its weight in 5 hours. This is also not given so far, that is the absorption of water. This kind of absorption of water, it involves the two things, that is the osmosis and water potential. Osmosis is a process in which the solvent molecules move from the region of higher concentration to the region of lower concentration through semi-permeable membrane. The membrane which allows only solvent molecules is called semi-permeable membranes. The living membranes are the animal bladders, egg membranes, whereas the non-living is the parchment paper. That is also very important. So there is an experiment to demonstrate the osmosis. So that experiment is a funnel experiment, and for which how to prepare the egg membrane which acts as which is a semi-permeable membrane. So for this preparation, you have to remove the yolk and other parts, making hole in the egg, and keeping the calcareous porous shell has to be placed in dilute HCl. This dilute HCl dissolves the calcareous porous shell, and the egg membrane remains intact. This is a semi-permeable membrane. Take thistle funnel and invert it and tie this kind of membrane, egg membrane, which can be prepared by using the dilute HCl. And this is the semi-permeable membrane. Pour sugar solution into this and note down the level of the sugar solution and place it in a beaker containing water and fix this to a stand. After some time, rising the level of sugar solution can be noticed. This is because of osmosis. Osmosis is a process in which the molecules, solvent molecules, that is the water, moves from the region of higher concentration to lower concentration through semi-permeable membrane. Here the egg membrane acts as a semi-permeable membrane. Osmotic pressure is the pressure developed in a solution when it is separated from a solvent. Always it is positive, but osmotic potential is negative pressure. Osmosis plays an important role in the absorption of water into the plants. The water present outside is the pure water when compared to the water inside the plant body, and the cells present in the root hairs act as semi-permeable membranes, the cell membranes, and from the region of higher concentration, the water makes its entry into the plants, and it is the xylem which carries not only inorganic and sometimes the organic substances, and even the hormones are also carried, and hence it is not a purely water conducting tissue. It also transports some organic and also some salts like the phosphate, sulfates, etc., where easily movable are phosphate, sulfates, etc., and not that much mobile. Non-mobility can be seen in case of calcium because it is present in the middle layer as calcium pectate. So anyhow, so this enters into the xylem. Thus osmosis plays an important role in the absorption of water. Next to this is water potential. The chemical potential of water is called the water potential. Water potential of pure water at a standard temperature and pressure is always 0. Water potential has negative value. So it is the strength of water, potentiality of water. Water always moves from higher water potential to lower water potential, whereas the water also moves from low DPD to high DPD, that is diffusion pressure deficit. And then the water moves from higher water potential to lower water potential. So equation is given. Water potential is psi W equals psi S plus psi P. Water potential, and psi S here, it is the osmotic potential, which is always negative, as I told you, because of the presence of solute. The water potential will be lost, that is negative, and the plus psi P, that is the pressure potential. It is the contribution of the turgor pressure on the protoplasm of the cell. Turgor pressure contribution on the protoplasm of the cell is called pressure potential. It may be negative or positive. Anyhow, the water potential is negative. And here the differences between this the DPD and the water potential. DPD, water always moves from low DPD to high DPD, and here higher water potential to lower water potential, and it has positive value, it has negative value, and these are some differences between the DPD and the water potential. So this water which has observed, and then there are three types of solutions, that is also very important: hypertonic solution, isotonic solution. It is even given in the chemistry also. Hypertonic solution: if the concentration of extracellular fluid is more than that of the cell sap concentration, such extracellular fluid called hypertonic solution. When cell is placed in hypertonic solution, cell usually contains 0.9% of NaCl salt usually present. This is also very important. When cell is placed in such hypertonic solution, so water moves out of the cell because it is the higher concentration of water present inside the cell than the here the concentration of water will be less when placed in a hypertonic solution, and it becomes flaccid. In a flaccid cell, shrinkage of protoplasm takes place called plasmolysis. So here the thing is, the plasmolysis, during which the water is removed from the cytoplasm. After cytoplasm, water present in the vacuole is removed. This is also very important for your NEET examination. It is given in a flaccid cell. If the concentration of extracellular fluid is less than that of the cell sap concentration, that is called hypertonic solution. When cell is placed in hypertonic solution, water enters into the cell and makes the cell turgid. So in a turgid cell, the water exerts pressure on the wall called turgor pressure. This is very important. When deplasmalized cell is placed in hypertonic solution, it becomes turgid again. This is known as deplasmolysis. And then next to this, if the concentration of extracellular fluid is the same as that of the cell sap concentration, water will not move. Such a extracellular fluid is called isotonic solution. That is also very important. And the next to this, how the water, radial movement of water. There are two means of radial movement of water: apoplast and symplast. Apoplast, in which the water enters through the cell wall. This is also very important. Cell wall. Apo means free. Cell wall into intercellular spaces of the parenchyma cells. Here the water doesn't enter into the cells, but in the intercellular spaces. Next, the passage cell, that is the endodermal cells, which lie opposite to the protoxylem. Is endodermal cell, do not contain Casparian cells, but here it enters into the cell, hence it is symplast at this point. This is very important. And then the non-living protoxylem trachea, again it is apoplast. So this is about the apoplast movement, that is cell wall, intercellular spaces, passage cell, here symplast, and into xylem, that is apoplast again. Whereas coming to the symplast, the water enters through the root hairs, that is cell of root hairs, and inside it enters into inside the parenchyma cells along with the protoplasm, and then moves from one cell to another parenchyma cells in the cortical cells through plasmodesmata, and then passage cell, but enters into xylem. Here it is apoplast again, as it is non-living, which does not contain nucleus or protoplasm. This is very important for your NEET examination. But apoplast is fast when compared to the symplast. This is also very important. Well, there is one method called then the water enters by three processes. That is one is the cohesion, adhesion, surface tension, and transpiration pull. So the water which has entered the root has to reach the top. This kind of upward movement of water inside the plant body called ascent of sap that is inorganic translocation, but here it is not now inorganic translocation, it is some organic substances and these maybe hormones are present in the xylem. So anyhow, this upward movement of water inside the plant body takes place through the xylem, First, J.C. Bose proposed a pulsating theory, vital theory called pulsating theory, and according to this, it is because of the activities of the living cells. Even though the cells were killed, effect of sap continued, hence the vital theory of J.C. Bose has been discarded. The most accepted theory, according to some, it is because of root pressure. However, the root pressure produced will be less as the theory has been discarded. But about the 130 meters height, ascent of sap takes place even to 130 meters height. This is because of the transpiration pull and cohesion force. This is the most accepted theory. Transpiration pull and cohesion force theory proposed by Dixon and Charlie. According to this, there are certain things like cohesion, adhesion, and then the cohesion, adhesion, and the surface tension. Transpiration pull plays an important role in the transporting the water to the mesophyll region. So, transpiration pull and cohesion force theory proposed by Dixon and Charlie. According to this, the water molecules are coherent to each other, that is the attraction between the similar molecules, and they are also adhered to the walls of the water vessels, that is adhesion, cohesion and adhesion. And then, because of cohesion and adhesion, the water forms a column in the xylem. A continuous water column is developing in the xylem without any gap. And then, because of transpiration, that is the loss of water which takes place at the top, especially stomata, 90% of transpiration takes place at the top. Water concentration decreases. As the water concentration decreases, DPD increases, and water moves from low DPD to the increase in the DPD exerts pull on the water column, that is called transpiration pull, and because of transpiration pull, cohesion, adhesion, and even surface tension, water reaches the top even to the greater heights, that is 130 meters. This is the most accepted theory about the transpiration. And then because the uses, what are the advantages of transpiration? And the surface tension, as you know very well, it is the water molecules are attracted more towards the interior region than the exterior region. That is, even the polar molecule nature provides this case of surface tension in case of the water in plants, that is the surface tension, cohesion, adhesion, transpiration pull plays an important role in the upward transport of the water and other inorganic salts in plant. And the advantages of the transpiration, that is it is the transpiration, because of transpiration pull, the water and salts are absorbed and transported, and the water required for photosynthesis also, for which also transpiration plays an important role, and transportation of minerals also, and reduces the temperature, makes the plant body cool by 10 to 15 degree. It reduces the temperature, that is the transpiration, and makes the cells turgid, and only a turgid cell can perform all the activities, that is the advantages of this transpiration. So, transpiration means loss of water in the form of vapor through the aerial parts of the plant body. 90% of transpiration takes place through stomata, and 10% takes place through cuticular transpiration, and only 1% takes place through the lenticel. And stomatal apparatus consists of two kidney-shaped guard cells. Guard cells contain nucleus and chloroplast. Inner walls are thick and non-elastic. Outer walls are thin and elastic, surrounded by subsidiary cells. And there are certain microfibrils present inside the guard cells, cellulosic microfibrils, and Here, if they are longitudinally arranged, the moment entry of water into the guard cells will be delayed. If it is radially elongated microfibrils, cellular microfibrils, then the water entry will be very easy, and the guard cells, when becomes turgid, pulls back the inner thick non-elastic walls and the stomata remain open during this. And then the food substances, multidirectional movement of the prepared food inside the plant body is called translocation of organic solutes, that is the photosynthesis product are transported through the phloem. So phloem is the food conducting tissue for which the experiment is the gurgling experiment. So what here, a central portion bark is removed in a plant. We can see after a few days the accumulation of the food from the top. This shows that it comes to the phloem. It comes to the phloem from upwardness, that is towards the downwardness, that is from source to sink. And this kind of the translocation of solutes through the phloem, and then phloem experimented by the Munch, that is called mass flow hypothesis. In plants, the prepared food always moves in an en masse ductstorm, in an en masse from the source to sink, for which an experiment is conducted by taking two bulbs made up of semi-permeable membranes connected through tube P, highly concentrated solution in A, less concentrated solution when placed in trough containing water. Because of osmotic pressure, the turgor pressure, more water enters into the A, but A and continuous flow of solutes takes place until it attains the equilibrium. Whereas this is, he compared A with the mesophyll, B with the consumption root chain, and P with the phloem. And then finally, in case of mycorrhiza have hyphae structures, that is fungi, roots of the pteridophytes and gymnosperms get infected with certain fungi, say for example glomus, and this supplies nitrates, so phosphates, so etc. Whereas the plants also provide sugar and N-containing compound, nitrogen-containing compound, sugars and nitrogen to the this mycorrhiza, but the mycorrhiza provides water and minerals, moisture and some phosphates, etc. to the plants in which they attack, And then here the pinus seeds will not germinate without the association of the mycorrhiza, that is fungal infection. That is also very important. And increase in the concentration of the hyphae. It increases the surface area for the absorption. That is also very important. And one thing which I have forgotten, I think sometimes the food translocation, that is from the source to sink, from above to down, will be reversed sometimes. Say for example, during spring season, the refoliation takes place, the foliation takes place, for which it requires food. In case of some plants where the food is stored in sweet potato, all those things, underground modifications and something like that, and there, here, reverse, these sweet potatoes, they become the source, and sink becomes the leaves. Thus, the source and the sink direction will be reversed in case of some plants also. And then guttation is a process in which the loss of water, exudation of water takes place in the form of droplets, in the form of liquid, that takes place through special type of cells called hydrathodes. They are present in few plants like colocasia, etc. And guttation takes place both during day and night times. When evaporation is low, guttation takes place. That's very important. So hence, I hope, my dear students, I have completed this 11th chapter, and of course first chapter in plant physiology, successfully, I think, given importance to the questions which will be usually appear in your NEET examination. All the best, read well, please subscribe, and don't forget to subscribe for further chapters. Thank you.